Dear students, in this video we will discuss about the Ramachandran plot. Each amino acid has an alpha amino group and an alpha carboxyl group. Many amino acids join together to form a polypeptide chain, which is then twisted or folded into a secondary structure of the protein, in which there are alpha helix and beta sheets. In the polypeptide chain, the peptide bond is formed between the alpha amino group of one of the two amino acids and the alpha carboxylic group of the another. So during peptide bond formation, one molecule of water is released. This is called dehydration reaction. In this bond, there is a partial sharing of electrons between the oxygen of the carboxyl group and the nitrogen of the amide group, because of which the peptide bond becomes a partial double bond, and the characteristics of this bond is that the CN bond length in peptide is shorter than any other CN bond in simple amines. Peptide conformation in a protein is defined by three dihedral angles: omega angle of peptide bond, phi angle between nitrogen and alpha carbon, psi angle between carboxyl and the alpha carbon CC. So there are three dihedral or torsal angle between the two alpha carbons of polypeptide chain, due to which backbone of peptide is formed in any protein structure. See, the structure of the polypeptide is depend upon the rotation of these angles. Omega angle of peptide bond rotate only 0 and 180 degree. For this, it is rigid and does not rotate, as well as the atoms attached to it are in the same plane. Another side, the phi and psi angles rotate from 0 to 360 degree in the manner of plus 90 degree, plus 180 degree, minus 90 degree, and minus 180 degree. All rotation of the angles are always clockwise. The first form of polypeptide is cis or trans. If the omega angle is 0 degree, then both the alpha carbons R group will be on one side, which is called cis form. But in this form, the chances of van der Waals repulsion force are very high. Therefore, peptides in nature are in trans form, in which R group exists on the opposite side, due to which the omega angle is 180 degree. Let's discuss what are van der Waals forces. 5 angstrom is minimum distance between the atoms over which van der Waals forces attract. Below this, they will become repulsive. Psi and phi can rotate from 0 to 360. This does not mean that any angle is possible in the stable polypeptide. These will only be found in a particular combination to avoid the van der Waals repulsive and to be stable. To explain this, Ramachandran plotted the graph. So in principle, phi and psi can have any value between minus 180 degree to plus 180 degree, but many value prohibited by steric hindrance between atoms. Dr. G. N. Ramachandran made a plot on a graph that whenever phi and psi angles are in given value, then the structure is possible and it will form either alpha helix or beta sheet. It depends on the combination of angles. Now, this is the Ramachandran plot from which so many questions are coming in the competitive examination. He plotted the graph for L-alanine, which was later fitted to all the amino acid of the peptide. In this plot, there are the three color combination. Dark blue conformations that involve no steric overlap and thus are fully allowed. The median blue indicates conformations allowed at the extreme limits for unfavorable atomic contacts. The lightest blue area reflects conformations that are permissible if a little flexibility is allowed in the bond angles. Still, if you are having the confusion in the Ramachandran plots, let's discuss the Ramachandran plots again. Ramachandran plots are actual the combination of the phi and psi angle of rotation. The x-axis are showing the value of the phi degrees and the y-axis are showing the value of the psi degrees. Ramachandran plots are also showing the variety of the structure: collagen triple helix, right twisted beta sheet, parallel beta sheet, anti-parallel beta sheet, right-handed alpha helix. Left-handed alpha helix theoretically also predicted in the Ramachandran plot, but till now it is not observed in any protein. Two amino acids are unique in Ramachandran plots. Lots of questions are asking from these two amino acids. The small flexible glycine residues were excluded from plot because they frequently fall outside the expected ranges. Since it has only one H and R group, it can form many angle combinations that do not have steric hindrance. As well, it enables turns in peptide backbone that would not possible with another residues. Second unique and important amino acid is proline. Five-membraned ring of proline prevents rotation nitrogen alpha carbon bond with almost fixed phi angle minus 65 degree. Ramachandran plot predicting proline disfavors alpha helix formation. It is important. Proline lack amide hydrogen, so not involved in hydrogen bonding in helix. It is also called as helix breaker. So after theoretical discussion, let's see which kind of the question is coming in the competitive examination. First one, the Ramachandran plot graphically shows which combination of torsional angles phi and psi of amino acid residues contained in a peptide are possible. Examination of the plot below shows that only certain regions of the conformational space are permissible. Why are all the theoretical combinations of phi and psi not possible? The options are A. Two atoms cannot occupy the same space. B. The geometry of the peptide bond that links two amino acid residues restricts phi and psi angles. C. Beta sheets and alpha helices determine the allowed regions of conformational space. And D. The tertiary fold of the polypeptides restricts conformational space. Out of these four options, the first option, two atoms cannot occupy the same space, is correct option. Next question is from proline. If the pyrrolidine ring of proline is reduced to a linear form, the new amino acid will have. In this question, ring of proline is reduced and converting in linear form of new amino acid. So phi angle will be affected. First option is constrained phi than proline. Second, constrained psi than proline. Third, relaxed phi than proline. And fourth, unaffected phi and psi. Because ring is converting in the linear form, so phi angle will be relaxed than proline. Thanks for watching. For more information, subscribe, like and share this channel. You can contact for the CSIR NET Life Science or NEET quick notes with previous year solved questions.